Dr. Durr is a genius. He's brilliant, smart, great, wonderful. He's very well educated, and he's going to share with you some information I think is really pertinent to all of us, but it's particularly interesting for me just simply because I have a little bit of knowledge about the imports related to the various species that we raise and the limited genetics related to what is actually here. You know, when people talk about they have great genetics, sometimes they don't really know that there may have only been 10 of those animals ever imported. So it's interesting to hear kind of a little story about genetics and what's involved in wildlife. So with that, I'm going to introduce Dr. James Durr.
Well, thank you very much, Brian. It's very nice to be here. I think we tried to do this about 20 months ago or so, and we finally got to pull it off. So I've been looking forward to this for a long time. So following Joe's lead, I'll tell you a little bit about myself. I grew up in Oklahoma in a rural area, hunting quail and pheasants and turkey and whitetails. And for me, exotics were exotics. I never thought I'd ever have a chance to have anything to do with any exotic species. But I went to school in Oklahoma and I made it out to Sul Ross, got a master's, made it to Texas A&M, got a PhD. And my PhD is in genetics. I'm at the College of Veterinary Medicine, but I'm not a veterinarian, I'm a geneticist. And I do wildlife genetics. I teach at Texas A&M, I teach human genetics, pre-vet and pre-med students, and I do wildlife genetics research. So what I'm going to try to do today is talk a little bit about the kinds of research that we do. And as a professor, you know, I find it important before you give a presentation on something, you really need to define what you're talking about so that everybody's on the same wavelength, so that we all are talking about the same thing. Because you can talk about genetics, and there would be different opinions of what genetics is. I work with a lot of producer groups, with different species around the world, actually. And I hear people say, this place has better genetics than that place. Or, I've got a really bad problem with inbreeding because I didn't have as many calves this year. Both of those statements may be true, or they may not be true. You may not have any inbreeding problem. It may be something else. And maybe the person across the road doesn't have better genetics, he's got better nutrition than you do. And so what I do as a geneticist is try to find out, is there a genetic issue? Is there a genetic opportunity? Can we use genetic technology to do a better job of managing animals? So that's generally what we're up to. What I'm going to do today is give a couple of examples of how my laboratory at Texas A&M has used genetic technologies to learn things that there was no other way to learn with wildlife. I have worked with American bison for the last 25 years. I would say Texas A&M University, my program and others, probably has done more American bison research than any place in the world. And I'm going to give you an example from Wind Cave National Park that I don't think you're going to forget, because we learned some things there that we would have never known had we not used genetic technologies. I'm also going to give an example recently of a problem we had with a Sika deer, and whether the Sika deer was a hybrid or not, and whether it should be put in SCI records. There was a question about it. I'm going to explain to you what happened, and I'm going to explain to you what we found out using genetic technologies. And again, these are just examples. I have 50 or 100 more examples like this that we've worked on in my laboratory at Texas A&M, but I chose these two because they're pretty simple, they're pretty straightforward, and I think you'll see my point. And then finally, I'm going to talk about current resources we have for exotic wildlife. The kinds of projects I'm going to talk about here are because we had prior knowledge. We had a lot of information. But a lot of exotic wildlife, we don't have that kind of genetic data. And I'm going to talk to you about what our needs are and what our opportunities are. So genetics is the scientific study of heredity and the inherited traits among individuals. That's kind of the textbook definition of it. Wildlife genetics is the use of molecular tools to determine things like parentage in a population, inbreeding, whether it occurs or not, genetic diversity, how much genetic diversity is there, ancestry, for example, where these animals came from geographically in the world, individual identification. Joe talked about the loss of animals through theft. In my opinion, I'm a geneticist, every animal you have, you should have DNA samples saved on. People can cut off tags, people can change tattoos. You lose an animal and you think you see it somewhere. If you don't have a way to prove that's your animal, you're never going to get it back. All you have to do is pull out 30 or 40 hairs, keep them in a paper envelope, and you have a genetic sample from that animal forever at room temperature. Everybody should be doing that. We also use genetics to look at the genes involved in horn, antler, body size, or disease resistance, or in some cases, behavioral genetics. However, I want to introduce a term to you that you've heard, but you may not be an expert at, and that is genomics. Genomics is a branch of genetics. It's a very, very important branch. It focuses on the structure, function, and gene mapping and editing of genomes. I promise you I'm not going to talk about editing genomes here, but I am telling you right now it's a big deal. And I'm telling you right now it's going to impact your businesses. But that's not the topic today. Genomic technologies can provide the tools to do the genetic technologies above, parentage testing, individual identification, ancestry, and so forth. Genomics, basically, is sequencing the entire genome of an animal so that we sequence every single gene, all 20 or 25,000 in a mammal. We put them all in the right order, and we know every gene and we know every mutation in that animal. It's possible. We do it in my lab every single day with different species. So, Wind Cave National Park. One of my favorite places in the world. If you've been there, it's in South Dakota. It's at the southern edge of the Black Hills. It's very, very important geographic region for the Lakota Sioux. They feel like that their tribe, their humanality came from Wind Cave National Park out of the caves. It's a great place to visit. I went there in 1999 with a graduate student, and I collected DNA samples. At that time, we were collecting blood samples from every animal in the herd at their roundup. So we had every bull, every cow, every calf in 1999, and we assessed them for genetic diversity. We did a landmark study for the National Park Service with all of the National Park Service herds, including Yellowstone, with their bison. Wind Cave was part of that study. It was a very successful study. And we liked Wind Cave so much that she and I and other graduate students went back every year for 10 years and collected DNA samples from all the calves. That's a lot of work. That's a lot of DNA samples. But then in 2011, we got the money to parentage test all 10 years of those animals. Think about that. We had every animal in 99, and we got the calves every year for 10 years. And we knew every dam and every sire in that herd of 350 or 400 animals for a 10-year period of time. That was a lot of information. Our findings, I think, were unexpected. I was surprised, and so were the biologists at Wind Cave National Park. I will tell you, before we talk about the data, it changed how bison are managed at Wind Cave National Park and other federal parks. So we ask a lot of questions. I'm not going to go into the entire study, but just the stuff we're going to talk about here. How many bulls? Remember, I didn't mention it, but it was on the slide. There was a 1-to-1 ratio of bulls to cows at Wind Cave. That's their management. That's how they do it. You get culled at about 2.5 years of age, and if you get culled, you go out, you get sold, or you get given away. If you stay in the park at 2.5, you're there till you die. And you die of natural causes, okay? So it's pretty much how bison herds probably were in the past before humans intervened, pretty much. So we asked the question, how many bulls produce calves each year? 1-to-1 sex ratio, remember, of 400 animals or so. How many bulls never breed in this population? How many calves are produced by each bull? What is the age of the bulls that produces the calves? Okay, we answered all these questions before we did the study. We were wrong. Okay, everybody thought they had an idea. People who had spent their lives at Wind Cave said, these are the animals that are producing all the calves. These are the bulls. They, for the most part, didn't know what they were talking about. When we had data, it became pretty obvious. So I'm just going to give you a couple of examples. I kind of truncated this down so that the data were a little easier to handle. I just took the middle 5 years, 2002 to 2006. During that time period, 107 bulls produced 280 calves over this time period. There was an average of about 175 bulls that could produce calves in those years. So right off, 175 bulls on average, and only 107 of them produce calves. So there's about 65, 70 bulls out there in that 5-year period of time never produced a single calf. If you were in production, those things would be hamburger, if you knew it. But in a national park, that's not their objective. That's not the point. But you should be able to see right quick. I need to get rid of some, if this is your herd, I need to get rid of some bulls that aren't producing calves because they're fighting, they're eating grass, they're causing trouble, and they're not doing what they need to do. If we look at the number of offspring produced by each of these calves, it gets kind of interesting because this is a total number here. So of these 107, a little over 50 of these bulls only produced one calf in that time period. Somewhere around 15 or so, 16 of these produced 2 or 3 calves, and then it dwindles off, and a few produced 8, 9, 10, 11, or 12 calves. A few, 1 or 2 bulls produced that many calves. But remember, we know which bulls did that. We know which bulls in that herd produced 12 calves, 11 calves, 10 calves, and more importantly, we know which bulls produced no calves. Here's the one that really surprised me. So this is bull breeding age versus percentage of calves they produced in that time period. This is the age of the bull. This red line is the average through these 5 years. So you can see, starting with about 4 years of age, 5, 6, 7, 8, those guys produce, that group, that year class group produces the most calves. Then by the time they're 8 or 9, it starts to drop off. But there's even some bulls over 12 years old that produce some calves, upwards to around 5, 6, 7, 8%. Okay, so it's still going on. But here's your big reproductive group bison. Fine. But what's going on over here? 1.5 and 2.5-year-old bull calves are producing 10, 15, 20% of our calves, different years. What in the world? A 1.5-year bull calf would need a step ladder. You would think. I had a really smart graduate student. She brought this data to me. I said, this doesn't make any sense. It looks like young bulls start breeding by the time they're 3 years old, they forget how. And then they remember by the time they're 4 or 5. She said, I don't think that's what's going on. She had spent a lot of time in national parks looking at bison during breeding season, and she said, I think those 1 and 2-year-old bull calves live with the matriarchal groups. They're protected by their moms, their aunts, their female cousins, and they're breeding with those animals. And by the time they're 3 years old, a 3-year-old bull calf, they get kicked out of those matriarchal groups because they're like junior high boys. Nobody likes them. Sorry if there's any junior high boys in here. And then when they're 4, 5, 6, 7, they're mature, they can compete, they can breed, and they get and do the job. She said, let's test this. Let's take these 1.5 and 2.5-year-old bull calves, take the cows that they're breeding and producing calves with, and see if they're more closely related to those females than an average female from the herd. Guess what? She was exactly right. They were breeding with their female relatives. when they're 1 and 2 years old. What does that mean? That means there's a built-in 10 to 20% inbreeding in a naturally occurring, for the most part, bison herd. That's important. Inbreeding's not always bad. It is if it gets out of hand, but at some level it's not. It's different than what we expected. Historically, these bison herds do have an inbreeding that goes on in them. Think about this, guys. Without this kind of technology, we would have never learned this. This would have been invisible to anybody who paid as much attention as they could. They never would have figured this out. This information has changed long-term genetic conservation management policies in some private herds and some U.S. federal herds. As a matter of fact, the largest bison herds in the United States, the owner of the largest bison herds in the United States, keeps all of his bull calves in those herds until they're 3 years old. And then those bull calves are selected out and go to feedlots. The reason is he wants to conserve genetic diversity in those herds forever. And in order to do that, he wants the largest number of males breeding in his population that he can get. It is a strategy to ensure the long-term conservation of the species in private herds. And some national park herds have taken this up too and have changed their policies based on this because other national park herds have different culling strategies for their bison. And again, modern technology is the only way to find this kind of stuff out. So let's go to a different example, more maybe of a wildlife example. So I sat on the records committee at Safari Club International. I am their species identification expert, and I use genetic technologies to identify different species when there are questions. I'm sure many of you guys are probably members of Safari Club International and are familiar with the records committee and the record book at Safari Club. A few years ago, this Sika deer was submitted to the record book. The person who did it may be in the room, I'm not sure, but he is a Texan. This was a record Sika deer. The committee, including some guys that know a lot about these Sika deer, had some questions about this animal because if you know these guys, you know this is a pretty unusual looking animal. The question is this tine right here, this unusual tine along this main beam. There's an extra tine here too that's not that unusual, but this tine right here is unusual. And it adds a lot of inches to that animal and made it a record. So they approached me, said, can we do anything with this? I said, man, if I could get some really good DNA samples from it, I can try because there are some Sika deer resources out there. So we talked to the owner of the animal. The only thing that was available was that trophy, that shoulder mount. We drilled some areas from the antler, and we also got some hide from the back of the wooden plate. And I isolated DNA from it. It's not easy, but we were able to do it. We had to jump through quite a few hoops. We did. The low-hanging fruit for this kind of a species identification analysis, as many of you guys know, is mitochondrial DNA. It is a lot easier to work with than nuclear DNA, and sometimes it will tell you the difference in two different species. So the first thing I did was sequence its mitochondrial DNA in my lab. I thought, if this thing is not Sika mitochondrial DNA, we are golden. This is not going to be allowed. We sequenced it, and unfortunately, for me anyway, at that time, it had Sika mitochondrial DNA. Doesn't mean that it's not a hybrid. Because remember, mitochondrial DNA is only inherited from the mother. So everybody in this room has your mom's mitochondrial DNA. You females can pass mitochondrial DNA off in your egg cells to your sons and your daughters. But us males can't pass our mitochondrial DNA off, okay? So if this animal is a hybrid from the male side, we wouldn't detect it with mitochondrial DNA testing. I hope that makes sense. I searched the world over to find resources to look at the nuclear genes in this animal. And luckily, a lab in the UK had just published a paper on service genetics, including red deer and elk and Sika deer and a few others. And I contacted them and I told them what my problem was. And they said, we looked at some Sika deer in our study. It was primarily red deer, but we looked at some Sika deer. Send us the sample and we will test it for the genes that we have determined tell the difference in these species. So we looked at a particular kind of marker. It's called a microsatellite marker. They looked at the marker. And they discovered numerous alleles that were common in red deer, but they had never seen in any Sika deer anywhere in the world. That led me to believe that this animal was a hybrid. The most likely explanation was that this animal was a cross between a Sika deer hind and a red deer stag. But it's not that simple. The original hybridization event had to happen at least 3 generations ago because it's not 50-50 red deer, Sika deer. It's about 5/8 Sika deer, 3/8 red deer. So the math's not going to work out. This isn't a first generation hybrid. This is a 3rd generation, 4th or 5th generation hybrid animal. So this animal most likely was produced 3 or more generations in the past. Female offspring from subsequent matings were red deer or I believe red deer, Sika deer hybrids were the males. Its father, its grandfather, or beyond were also hybrids until the production of this particular animal. This was a complicated case because it's not a straightforward first generation hybrid, but it is a hybrid. This explains the presence of Sika deer mitochondrial DNA because it's got a direct Sika deer lineage. All the females in its history are Sika deer. Just the males that were brought in were hybrids. The exact number of generations, I can't tell. It doesn't matter. The only question we ask is this Sika deer or is this not? And it's not. It's a hybrid. The Records Committee rejected this thing. And again, if you're the guy who submitted it, I'm sorry, but it happens. So those are just 2 examples of how we use this technology. Again, I could give you a lot more examples. We do this stuff all the time in my laboratory. We use technology to answer questions that there's no other way to do it. And it's really fun and it's really exciting. And it's like a fishing trip every day. You don't know what you're going to catch. Let's talk a little bit about current genetic resources for exotic wildlife, the stuff you guys are interested in. Worldwide, what genetic tools exist for these wildlife? Well, you can go on the web, or you can go to my website, or you can go to any place, University of California, Davis, or other private companies here in Texas and Oklahoma and other places, and you can look and see what services they provide. And I just listed most of the species that I know of that are commonly provide parentage testing, individual identification testing, and so forth. Those are pretty much the species that are off the shelf. There's test for that's been developed. The groundwork has been done. The tools are available. You can go do those tests. I can do them in my lab for virtually any of those species. Our understanding about these species has been significantly benefited from genomic studies, sequencing, gene identification studies that researchers have done. Many of these researchers you guys don't know anything about. They're off in Europe, or they're in Germany, or they're in South Africa, Stellenbosch, or the vet school in South Africa. They're doing this kind of research. They're not doing it for the wildlife industry. They're doing it for long-term wildlife conservation studies. But most of these U.S. exotic species that you guys are interested in, we have very, very little genetic or genomic resources for. I say let's change that. We need to develop genetic tools to consolidate the known genomic information for a number of species. And I'm not going to read off all these species. You guys know these things. But these deer species right here, every one of these deer species here may have some genomic data out there if you search around, but for one animal and a partial sequence. And it's real limited. And very little or no parentage test or DNA identification test. They're just not available. For the antelope species, many of you guys are interested in probably virtually all of those antelope species. If you call me Monday morning and you say, I need to do parentage testing on my whatever, I'm going to tell you, I don't have the resources to do it. There's no off-the-shelf kit that I can go buy and go do parentage testing for you for these species. We recently had a case where within the last week or two, a person contacted us and wanted to be able to tell the difference between a red and a Kafui lechwe. Can we tell the difference between if you know the lechwes, you know why. There's a big difference between having a red lechwe and having a Kafui lechwe. You know, can you tell me genetically? Can I send you some DNA and you tell me which it is? And the answer is no, I can't. It would take a tremendous amount of research for us to do that, and more than you're going to want to pay for. We can't do it. We can't do it reasonably anyway. And I mean, somebody may tell you they can do it, but I would suspect they can't. Wild cattle, you may not worry a lot about some of these, but certainly Cape buffalo. Cape buffalo, there are some resources for that have been developed in South Africa. We do have some genetic resources clearly for them, but most of these others we don't have a lot. Sheep species too, I've talked with Wild Sheep Foundation folks a lot about developing these resources. They would love to, but we just haven't been able to get the financing together to pull it off. And along with that, the goat species. And then plenty of other species. There are some resources available for giraffe. There are none available for okapi. Plenty of resources from Stellenbosch in South Africa for rhinos. Plenty of resources available for the equids, including zebras. There's some predators that people at least raise. And for some of those predators, there's resources. Some of those, there's not. It's pretty spotty. For most of these species, we have little genetic tools available off the shelf. My hands are somewhat tied because we simply don't have the knowledge base to do the things that we need to do simple parentage tests for you. You got 3 bulls, and you got 12 cows, and you had 8 calves. Which of those 3 bulls is producing the calf? And we can be talking about bongos, or lechwe, or kudus. That's an important question. That could be a very important management question for you, especially if you have 3 ugly calves and 5 really great calves. You want to know, those 3 ugly calves have the same dad? And we're having him tonight for dinner. We always talk about loss of genetic diversity and inbreeding in exotic wildlife in the United States because of a founder effect, because there were few founders brought over from the natural populations. We always talk about it, and it's probably true, but it's not documented. It's not documented with the kinds of technologies that I'm talking about here, and that's what we need. We need genetic documentation so that we know where to go and get the genetic resources in Africa or India or Asia or Pakistan or wherever we need to go. Where are we lacking that genetic diversity? Where's it from? Genetics can tell us that. And finally, as I mentioned, individual identification is important. And I would stress, if you don't take anything else from my presentation, you should be collecting genetic samples. Keep them in your safe at home. You can keep them at room temperature. Send them to me at Texas A&M. I have an archive. I'm going to talk about that in a minute. We'll archive the samples for you. And if something comes up, you lose an animal, and you need to prove that's your animal, and Texas A&M has your hair sample that you collected 3 years before, you can prove it if we have the resources. I think we need to consolidate all of the known genetic resources and genomic resources into one database. There's no database in the world that has all of those resources consolidated. We need to identify species with the greatest need for targeted genomic sequences, and we need to find the funding to do it. We need to start sequencing these things, producing and developing the tools we need for this industry. We need to develop, first and foremost, the low-hanging fruit is a mitochondrial DNA-based species identification test that can be used off the shelf, where you could send me a meat sample and not tell me anything about it, and I send you back the name of the species, and do it with mitochondrial DNA. Hybridization causes a problem, as I've shown, but most of the time, mitochondrial DNA does an excellent job for species. And we can do that. We can do that pretty simply and fairly cheaply. More importantly, though, we need to develop nuclear genotyping microarrays. And I'm not going to go into what microarrays are, but they allow us to look at a lot of genes at one time, thousands of genes at one time, for different species. We could develop an industry-wide microarray gene panel for all of the exotic species that you're interested in. One single resource that would identify all of these markers, parentage, ancestry, inbreeding, for all of these different species. It's possible. In fact, these tools can be used to evaluate and better manage species. We can tell what country of origin they came from in some cases. And worldwide conservation efforts could also be improved. If we develop these resources here for kudu, then we can use those same genetic resources in Namibia to help better manage kudu there. So I'm not narrow-minded enough to think this is only a Texas-centric kind of thing. We always have to think worldwide. We always have to think how our actions can impact these species worldwide. And I'm talking about developing worldwide resources for the management and conservation of these species. The College of Veterinary Medicine, which Dr. Cook, you're going to hear from this afternoon, and I are both faculty members in, has a talented group of geneticists and genomics researchers that are interested in developing these technologies. In fact, Texas A&M and the College of Veterinary Medicine houses the Exotic Genome Repository. I know, Brian, you've talked to Brian Davis a couple times about this. He's in our college. He's our colleague. I work with him on a daily basis. That's the website where you can go and look at exoticgenome.org. He has tens of thousands of reference DNA sequences from exotic species in his website. He works primarily, however, with zoos, not so much with this industry, but he should be, and he wants to be. And he actually really wanted to come to this meeting, but he had another commitment. In addition, in the College of Veterinary Medicine, we have the largest collection of genetic samples for any wildlife species, over 80,000 DNA samples from North American bison. These samples were collected from every major bison herd in North America and Canada and Mexico. I don't think there's any other species on the planet where we have collected, archived more than 80,000 animals. It is a huge collection in my lab, and we've been collecting these things and doing the research with about 6 now PhD students over the last 20 years. And finally, researchers in the College of Veterinary Medicine have sequenced and characterized genomes from 50 or so that I could just count mammal species over the last 10 years. I'm telling you, I'm real Texas A&M centric, but I'm telling you, Texas A&M University has some of the finest researchers in this area in the world. And you can talk to anybody in any university and ask them about the researchers in the College of Veterinary Medicine at Texas A&M for these kinds of resources, and they'll tell you Texas A&M is a leader in this field. We have this university here. Dr. Cook and I are part of it. And the reason we're here is because we would love to work with you. We would love to start putting together these resources for exotic wildlife. I think what I've tried to do is show a couple of examples of how we've used resources to answer questions. And I've also tried to highlight the areas where we need more information. We need the ability to do genomics work with a lot of these species. And it would not only help this industry, we could help wildlife conservation efforts worldwide for these species if we were involved in this. So I'll be here all day, and Dr. Cook will too, and we're happy to talk with you about this and what we might be able to do in the future. Thank you very much.
That was excellent information. I hope everybody was able to gather some conclusions about, again, why we're here today. You may or may not know it. He talked a little bit about the zoos here in the United States and some of the work that they do. In the Association of Zoos and Aquarium, which is primarily made up of city zoos, public city zoos, they work as a group together. There's a little over 200 zoos that are part of the program, and they have what is referred to as the Species Survival Plan. And essentially what they do is they share information with one another, and they do pretty extensive research to ensure that the diversity within the population that they have in the zoo is sustainable for the future. And they classify each of the different species based on colors. But the goal is that zoos don't sell tickets because they have taxidermy. They sell tickets because they have live animals in the zoo. And importation of a lot of the species that are in the zoo is virtually impossible. So the only way to make sure that they have the ability to have their product for sale in the future is to ensure that they have diversity within their populations. And the reason that I bring that up is that as an industry, from my perspective, we have very much acted like hobbyists rather than serious business people. And I don't want to use the word irresponsible, but our practices in many cases are not conducive to the long-term viability of the species that we're raising. And with what these animals cost these days, what I'm seeing is people are coming together and they're starting to view this differently than as a hobby or just as a pastime, but they're viewing it as a business. And with Texas A&M being here in the state and their openness and willingness to work with us and to assist us with what we're doing, this is not just about producing big horns or a trophy animal. It's about ensuring the long-term viability of the species that we're investing our money and our time and our ranches in. So I would really like, our company, we catch annually somewhere between 3,000 and 5,000 animals per year. And we've just implemented a policy this year where we are taking hair samples on every animal that we catch. And we're taking these hair samples, we're submitting them to the EWA, who is starting a registry for several species. But we're just developing the practice of doing exactly what he said, not necessarily for the purpose of theft, but we believe that by gathering data and submitting the data to the proper resources, that it will ultimately prove to be very beneficial, not just for us, but for everybody. So I would ask all of you, if you have animals and you are moving animals, or if you have the opportunity to gather hair samples, please do what he said. Get those samples and keep them yourself or submit them to the university. But the more data that we can gather as a whole, the better off we're all going to be. So thank you very much for your presentation. It was very useful, and I got a lot from it.